Okay, so we are ready to start looking at our second lecture about cellular respiration. This one ideally will be a shorter lecture, however, it will be slightly more complex. So first off here, we started talking last time about the different stages of cellular respiration. Our ultimate goal was to break down a big organic molecule like glucose or some other, like a lipid or something, and get a lot of ATP out of it. Our first stage was glycolysis. That'll be followed today by the Krebs cycle and an electron transport chain. Those are going to look really, really similar to some of what we had seen in photosynthesis. So first off, glycolysis. Where does it happen? It happens out here in the cytoplasm. What does it make? It makes some little bit of energy, but not enough in the form of ATP. But we still have this 3-carbon pyruvate molecule that we need to fully break down and extract all the energy from. That's going to be the process that's going to occur in the Krebs cycle and then the electron transport chain. But to do the Krebs cycle and the electron transport chain, we have to have oxygen available to us. So that means that those processes are aerobic. Glycolysis is anaerobic and does not need oxygen to make it happen. However, the Krebs cycle does. Once again, this is why you and I have to breathe oxygen. If we do not breathe enough oxygen, we cannot keep the Krebs cycle running and we cannot make enough ATP to keep our cells alive. So glycolysis, we lysed a sugar or we split glucose in half and we got a little bit of ATP from it. So now we're ready to move on. We've already broken apart our glucose, but we still haven't used our oxygen. Ultimately, we're going to break down our glucose molecule fully, make some CO2, make some water, and generate quite a bit of ATP. So to do that, to continue doing that, we need to take our pyruvic acid. As you can see here, our 3-carbon pyruvic acid. It's still carbon bonded together with, there's oxygen and hydrogen as well. We can disassemble that and get more energy out of it. So the Krebs cycle, sometimes called the citric acid cycle, is really very similar in some regards, totally different reactions, but similar in nature to the Calvin cycle. In the Calvin cycle, we grabbed carbon from the atmosphere and stuck it together using energy. Well, this is the reverse of that. The Krebs cycle takes energy stored in carbon bonded molecules and it breaks it apart in order to get the energy back out. So it's like a reverse Calvin cycle. Instead of taking in carbon dioxide, it actually gives it off. This is why you and I right now are exhaling carbon dioxide, because this is happening inside of our bodies right now, and we're ripping apart some food that we ate earlier in the day or yesterday or whatever, and we're exhaling that carbon dioxide. Okay, so here's how this works. We grab our carbon molecule, that pyruvate, we break off a carbon. In doing so, we get some NADH. Remember, NADH is holding an energized electron for us. We then put that 2-carbon molecule into a cycle. Okay, just like in the Calvin cycle, we had to grab that carbon from the atmosphere to get it into the cycle. This has a molecule that grabs this 2-carbon molecule and brings it into the cycle. Basically, we're able to chemically bond to that molecule and start ripping it apart further. So it forms temporarily a molecule called citric acid, which is, yes, it's citric acid like you guys might know about. That's why it's called the citric acid cycle. But then right away, we tear off a carbon from it in the form of carbon dioxide, and you exhale that. But in doing so, you charge up some NADH. You do that again, and you tear off another carbon, and you make some more NADPH or NADH. You then rearrange it, and you get some ATP. You rearrange it again, and you get something called FADH. Guess what it does? Yes, it holds electrons. You regenerate the cycle back to its original state, and then you get some more NADH. And then you're ready to grab another molecule of this acetyl-CoA and start ripping it apart again. So it's a cycle. It goes over and over and over again. As long as you have oxygen to help keep this running, and your broken apart glucose the pyruvic acid stuff, you will constantly keep the cycle running. You'll give off carbon dioxide. You'll make a bunch of NADH, and you'll make some ATP. Keep in mind that one molecule of glucose can turn this twice. And so you get actually quite a bit of this NADH stuff and a little bit of ATP. And you give off quite a bit of carbons, right? 6 carbon dioxides total, because you totally disassemble that glucose molecule, which is C6H12O6. All right, so the primary idea here is the Krebs cycle disassembles those remaining carbon molecules and extracts energy from them. That energy is being held primarily as energized electrons in this NADH and FADH. Just like in photosynthesis, we stored electricity or energy, not electricity, electrons inside of our NADPH. We're using a very similar molecule, but we're doing the exact same thing. Okay, so we made 2 ATP, 6 NADH, and 2 FADH2s. ATP is instant energy. We also made 2 ATP during the glycolysis. That's 4. We want to make 36. So we've got 32 more ATPs yet to make. How are we going to make that? We need to have something to power the generation of ATP. And so what we're going to do is exactly what we did in photosynthesis, which is use an electron transport chain. So NADH and FADH2 are both carrying high energy electrons. Those high energy electrons get passed through this electron transport chain. So where are we at right now? We're in the mitochondria. And in the mitochondria, we have a membrane. The mitochondria has an outer membrane and an inner membrane, just like we saw in the thylakoid membrane in the chloroplast. Embedded in this membrane are membrane pump proteins. And those membrane pump proteins, as they have energy from energized electrons, they pump hydrogen, right? Same thing. Hydrogen gets moved across the membrane and it pumps an artificially high concentration gradient up here. We know it wants to fall back from high to low, and in doing so, we can use it to power the generation of some ATP. But we need some protein or enzyme that can do that for us. Once again, that is the ATP synthase molecule. Just like in photosynthesis, when that hydrogen falls from high to low, it's able to turn this protein pump, ATP synthase, which can attach that third phosphate group onto adenosine diphosphate and convert it to triphosphate. So as long as you're dumping in NADH and FADH and you're providing all these electrons are constantly pumping hydrogen, and in doing so, it's turning this turbine, this biological turbine, to recharge ATP. So that's how your mitochondria actually acts as a power plant. It's almost like your mitochondria is like a hydroelectric dam, right? You put water behind it, it turns turbines and makes electricity. But in this case, you're putting hydrogens inside of it, you're packing it full, and then as those stream back out, they turn these turbines of ATP synthase and generate ATP. So your total ATP, glycolysis, you got 2, Krebs, you got 2, but the electron transport chain, you got 32. But remember, that came from that NADH and FADH that you got earlier. And that gives you in total 36 ATP to keep yourself alive. So if you think about that one little molecule of glucose, you're consuming lots and lots of molecules of glucose every single day, thousands and thousands and thousands of them. And you're generating so much ATP, like pounds and pounds of this stuff, but you instantaneously use it. It only lasts for 2 minutes. So if you don't use it right away, you lose it. So you're constantly making it and then using it to power your cells, recharging the ATP, using that whole cellular respiration thing over and over again. We have to keep that running. You have to have food, like glucose, and you have to have oxygen so you can fully break down that food. As long as you have oxygen and food, you can keep yourself alive. If you lack either of those, we know that doesn't work out very good. But this is why you have to do this. This is also why you exhale your carbon dioxide.